A very good afternoon to everyone. On behalf of Walters Kluwer, a warm welcome to our speakers and attendees of this microbiology webinar under our MedicalEdge series. Our today's topic is bacterial growth, nutrition and metabolism. Our speaker today is Dr. Sumati Murlidhar, who is MBBS MD Diploma, Professor of Microbiology from Vardhaman Mahavir Medical College, New Delhi. She is also a consultant microbiologist and laboratory in charge, Apex Regional STD Laboratory and State Reference Laboratory of HIV, Southern Hospital, New Delhi. Also, we'll be taking all questions at the end of the webinar, and I request you to please post all your questions in the Q&A panel of this event. We'll try to address the maximum questions at the end. We'll also provide your feedback via link, which we will post in the chat section of this webinar. Now we begin our session. I hand over to Dr. Sumiti and I request her to please start sharing her screen.
Yeah, good evening, everybody.
Ma'am, you can share your screen now. Thank you.
Is it visible?
Yes, ma'am.
I think I'll have to slideshow.
Put in the slideshow, ma'am. Yes.
Yeah. Yeah, good evening, everybody. At the outset, I'd like to thank the Walters Kluwer team for putting up this show today. And this topic, that is bacterial growth, nutrition, and metabolism, it's one of the initial topics in the textbook of microbiology. As students, all of you must have seen that the first few subjects, chapters are on morphology and physiology of bacteria. So there is a reason why I selected this topic. That is because it's one of the early chapters and one of the chapters that will lay the foundation for your knowledge in microbiology. So if your basic knowledge of microbiology is strong and a strong foundation is laid, then you can you will be able to understand things better later on when you move on to other chapters in microbiology. And at the outset, I would also like to tell you that most of the contents of my lecture today, they have been taken from our textbook, that is the Lippincott's Illustrated Reviews, the textbook of microbiology, which has been written by Dr. Rohit Chawla, who's my co-author, and me. In fact, both of us are teachers in medical colleges. He's also a professor in Delhi, and I am also a professor. So we have, with a finger on the pulse of the students, we have written this textbook so that it covers all aspects in the way it has to be learned. And also we have kept in mind the CBME and written the book according to that. That is a competency-based medical education assessment, as it is required now for as per the the Council of the National Medical Council. So with this, I'll begin my topic. Okay. So microbiology, the subject of microbiology is a very colorful one. So there are some reasons for it. One reason would be that most of the medical microbiology departments in most medical colleges are predominated by the ladies. So you will see a lot of colorful attire in the department because there are many ladies. Secondly, the bacteria themselves are very colorful. The bacteria and other microbes, whether it's bacteria, the fungi, many of them are pigment producing. So you see a lot of color in this department. Apart from color, you will also see a lot of shapes and sizes of bacteria and microbes. So they are known for being varying in shapes and sizes. And where do we find these microbes? They are ubiquitous. Ubiquitous means they are found everywhere. You go to the mountains, you will see different types of bacteria and other microorganisms. You go to the depths of the seas, you will see. And you go to the coldest places, the hottest places on the planet, everywhere they are present. And also in human beings. So even in us, in all of us, the oral cavity, the gut, they are all full of bacteria and other microorganisms. So you cannot escape the microbes from anywhere on this earth at least. So they are present everywhere. So now today's topic is physiology. Of course, the other important topic is the morphology, which is about study of the structure and the shape and size and all of that. But that is not our topic today. Today, I will be talking about physiology of bacteria. So physiology involves the growth, nutrition, and metabolism. So when I say growth, it means an increase in size. And it's not an increase in number. So mind this, bacterial growth refers to an increase in size only. When it is an increase in number, you call it reproduction or bacterial cell division. So bacterial cell division, I'm sure all of you from your biology classes in school, you must have heard that bacteria, they multiply by binary fission. That is how they divide and reproduce. So binary fission, that is bacterial cell division, this happens by binary fission, that is binary refers to two, and fission is divided into, cut into two, you can say. So the cell wall, the cell membrane, the chromosomes, all of these divide, and that's how bacterial cell division occurs. Firstly, the cell membrane divides, and then the cell wall and the chromosomes. And finally, one bacterial cell gives rise to two, 2 to 4, from 4 to 8. So that's how doubling populations occur, and exponentially the bacterial division occurs. Now, when the bacteria divide, they tend to, the daughter cells tend to stick together and form certain shapes. So that gives rise to the arrangements of the bacteria, whether they are cocci or bacilli. I'm sure by now, after your morphology class, you would be knowing what cocci and bacilli are. The round-shaped bacteria, spherical, they are called as cocci, and the rod-shaped or the longer ones are called as the bacilli. Apart from that, you have the spiral bacteria. So when the bacteria divide, the daughter cells stick together and they may form certain arrangements. Like if they are in the form of pairs, you call them diplococci. In the form of 4 tetrads and 8 sarcina. And if they are in clusters, you call them staphylococci. Staphylos means a cluster of grapes. And if they are in the form of chains, you call them streptococci. So similarly for the bacilli. So the arrangement depends on how the daughter cells are arranged after cell division. Now coming to the generation time. What is generation time? It is also called as the population doubling time. That is the time required for one bacteria to give rise to 2 daughter cells. Okay, so the time it takes for the bacteria to double its population is called the population doubling time. Now let's see why this is important. The generation time is different for different bacteria. For example, if you take the Escherichia coli and other medically important coliforms, most of them, the generation time is around 20 minutes. That means it takes about 20 minutes for the population to double. And if you take Staphylococcus aureus, which is another very important bacteria causing several infections, it's also around 20 to 30 minutes. But when you take Mycobacterium tuberculosis, this is the bacteria which causes the disease tuberculosis. This takes about 20 hours to double its population. And then you have Treponema pallidum, which causes one of the sexually transmitted diseases, that is syphilis. It takes about 33 hours for doubling its population. And if you take the leprosy causing bacteria, that is Mycobacterium leprae, it takes about 20 days. Mind you, this is not in minutes or hours, it's in 20 days. So you have bacteria which multiply and double their population in about 20 minutes, 20 hours, 20 days. So why is it important to know about this? Because in the laboratory, when we start growing the bacteria as a diagnostic procedure, and when we culture them in the laboratory, we need to know their requirements of the time it requires for them to double and multiply and all of that. Because, for example, Mycobacterium tuberculosis, it takes about 20 hours, which means that for a colony to form, it may take anything between 4 to 6 weeks for visible colonies to be formed in the laboratory when you're culturing them. So when a patient comes to you and gives his or her sputum sample for culture of Mycobacterium tuberculosis, you cannot tell them that you will give the report the next day. For other bacteria, you may be able to give the report the next day, but for tuberculosis, since you have the knowledge that it takes about 4 to 6 weeks to grow, you will be telling the person that the reports will only be available at the end of the month. So these are important points you have to know, and that's why it's important to know the generation time of bacteria. Now, the bacteria that grow in the laboratory, they grow in the form of, in many forms, depending on the kind of media you use. So if you are using a solid media, that is, you're providing an artificial medium for the bacteria to grow. If it is a solid medium, they form colonies. Colonies means the growth of the bacteria. They are called as colonies. I'll show you a few figures later on. So they are formed by growth of the bacteria on the solid medium. Whereas if it is a liquid media, how do you assess that the growth of bacteria has occurred? That is by looking at the turbidity. So when there is a lot of bacterial growth in liquid media, you see that the tube will be turbid. So that will indicate the growth has occurred. And the third is biofilm formation. So on solid media, this is what it looks like. These are the plates that we use in the laboratory to grow the bacteria. So we use different kind of culture media. They're called, culture is growing the bacteria, and the plates on which we grow, we call them the culture media. So all these dots that you see, they are the colonies of bacteria. So if the colonies are plenty, it forms a confluent growth, as you can see in two of these figures. And the red plate is because it is blood agar. So depending on what kind of media you use, different bacteria will grow on them. And this is what it looks like in a liquid medium. On the right side, you can see that the liquid is clear. That means no bacteria has occurred. But on the left, you can see that the liquid has turned turbid. So after incubation, if the liquid turns turbid, it means bacteria has grown in them. And what is a biofilm? It is nothing but a community of bacteria or microbes which are embedded on an inert or living surface. So here, a community of microbes means all of them are usually covered by a matrix or some organic polymeric substance called as the glycocalyx or slime, and they form a biofilm. Usually this happens on an inert surface. Now, coming to the kinetics of bacterial growth. So when you add one bacterium into a suitable liquid medium, what happens? If you add it in a suitable liquid medium where there is enough nutrition for it to grow and all other conditions of growth are fulfilled, and when you incubate it at appropriate temperature, the bacteria will grow, and the bacteria counts are made at intervals after inoculation, and this can be plotted in a graph that is called as the bacterial growth curve. So how does the bacteria grow? What are the phases of growth? Basically, there are 4 phases: the lag phase, the log or exponential phase, the stationary phase, and the phase of decline. Now, the lag phase is there is no increase in number here. So when you just add the bacteria, the first phase is the lag phase. Here, the bacteria will not immediately start multiplying. It requires a little time to get used to the environment that it has been put in, and it requires some time to to assimilate all the nutrients and then start growing. So that phase is called as the lag phase, where it is getting ready to grow. And the log phase or the exponential phase is where the cells start dividing and their numbers start increasing exponentially. So this is where the bacteria grows in number. And the third phase is the stationary phase, where the cell division, that is the multiplication of the bacteria, stops because of the depletion of all the nutrients that you have given in the media. And many toxic metabolites will start getting accumulated. So the bacteria stops growing here. That's why it's called as the stationary phase. But this is a stage where equilibrium exists between the number of dying cells and the newly formed cells. So basically the count may remain the same, because as the bacteria are dying, they are also new ones are being formed. So stationary phase is where it is stationary and the graph will plateau off. So that's why it's called the stationary phase. And phase of decline is when the population of the bacteria decreases due to death of the cells. Okay, so here I want you to remember that the word stationary is spelled as A-R-Y. Please remember, it's not E-R-Y. The stationary that is E-R-Y refers to your, pen, pencil, eraser, and all of that. So it's not that stationary. So this is stationary phase. And this is the bacterial growth curve. What I have explained now, that is the lag phase where the bacteria is getting ready to multiply and all the nutrition is being taken in and it's growing, and then it will multiply in the log phase. And finally, the stationary phase where it just plateaus off, and the phase of decline where the count reduces. You must be seeing there are two kinds of lines here. One is the total count, which does not reduce, which remains the same. And the viable count is the one which falls. So I'll tell you more about this a little later. Now, this is a very important part of your lesson, because many of your exams, one of the theory questions, it's a very important theory question where this may be asked. Draw the bacterial growth curve and label its parts and describe the various phases of bacterial growth. So this is an important question from your theory point of view as well as your viva. Morphological and physiological alterations during the growth. So this is very important. This again may be asked in the exams. The lag phase is when the maximum cell size occurs towards the end of the lag phase. So I told you during the lag phase, the bacteria is growing slowly and it's increasing in size, but not increasing in number yet. In the log phase, the cells may be smaller because they are multiplying very fast. So they may be smaller, but they stain uniformly during this phase, and the numbers are increasing exponentially. Then during the stationary phase, there is irregular staining. So when you do a gram staining in your practical classes, you will see that if you are doing it in the log phase, the staining will be very uniform and beautiful. Okay, the stationary phase, again, this is the phase where sporulation occurs and all the antibiotics are produced and exotoxins are produced. This again is important from the applied aspects. And phase of decline, where you get involution forms, that is with aging, the bacteria starts disintegrating. And that's when the phase of decline happens. And bacterial counts, coming to ways of counting bacteria. There are two types of counts, bacterial counts. You saw the graph just now, which said the total count it showed and the viable count. Now, the total count gives us the total number of bacterial cells. That means it includes the living cells as well as the dead cells. So when you come to the viable count, it is only the number of living cells. So both are important in a way. So how do you count? You count the bacteria by many methods. One is the direct counting under a microscope using a counting chamber. Or you can use electronic devices such as the Coulter counter. And the direct counting can also be done by staining the smears, make a smear of the bacterial culture, and then you count the stained smears with a known volume of the culture. Or you compare the relative numbers in smears with known number of other cells in mixed culture. Or you can use an opacity measurement tube. I'll show you what it is. Or you can separate the cells by centrifugation or filtration, and then you measure their dry weight or wet weight. Or chemical assay of the cell components such as nitrogen. So there are many ways, methods. We don't usually do this in a diagnostic microbiology laboratory. It is more for a research purpose. So it's not that important for you to know, but you just have to enumerate some of the methods if you are asked in the viva. Now, counting chambers, this is one way of counting. You must have seen in your physiology classes, you must have seen in your first year subject, the Newbar's counting chamber. This is what it looks like, the hemocytometer. And in that you can, just as you count your RBCs and WBCs, the way you have been doing it in your class in physiology, similarly, you can do the counting of bacteria if you know the amount of fluid that you have taken. So you do the calculations just as you were doing it in physiology. And turbidity, this is another way of counting. You use a spectrophotometer where the amount of light that is passing through the tube, that is with the turbidity. So the light scatter by the bacteria will, that is measured, and then the counting is done. These are automated procedures. Or you can also use a McFarland control tube. These are control tubes where a given amount of bacteria are there in each of these tubes. That is, usually it is 1 to 10. Tube number 1, 2, 3, 4, it goes like that, and with increasing turbidity. So if your bacterial culture turbidity of the tube compares with, say, number 0.5 or 1 or 2, then according to that, the count is given off by reading of the turbidity of the McFarland tube. So these are the control tubes that we use. Now coming to viable cell, this was about the total count. But when you come to viable count, how do you measure that? So it measures the number of living cells. So how do you do it? Either by dilution method or by the plating method. Here you count the number of colonies that develop after incubation, and it gives an estimate of the viable count. So how do you do it? You originally start with, say, 1 ml of the culture, and then you add 9 ml of broth. So total you have diluted it 1 in 10 times. So 1 ml of that you put into another 9 ml broth, and that will become a double dilution, that is 1 in 100. So you go on adding 1 ml from each of these tubes to the next tube till you get a dilution of, say, 1 in 1 lakh. So you have 1 in 10, 100, 1000, 10,000, and 100,000 or 1 lakh. So now 1 ml of each of 0.1 ml from each of these tubes, you plate them on the culture plates. And then you incubate them and you count the number of colonies. For example, in this, you find that 6 colonies are there in the second last tube plate, and the last plate at the bottom has 0 colonies. That means nothing has grown in the 1 in 1 lakh dilution, but in the 1 in 10,000 dilution, you have got 6 colonies. So you count the colonies and then you do the calculations because you have started with 1 ml. So those were the ways of counting live bacteria and the total bacteria, that is the live and dead bacteria. Now coming to the nutritional requirements of bacteria are similar to that required by humans and other life forms. Everybody requires, all living beings require carbon, nitrogen, inorganic ions, and certain growth factors. Why do they require all these nutrition? Because they, for generation of energy and for synthesis of cellular material. Now, chemically, the bacteria consist of basically 75 to 85% is water, which is in a bound form, and free water. And the rest is dry matter, which includes organic and inorganic parts, just like the human beings. So in organic parts, you have the proteins, the lipids, the polysaccharides, the nucleic acids, mucopeptides, and the mineral substances or inorganic part includes nitrogen, sulfur, phosphorus, calcium, potassium, sodium, magnesium, manganese, and all of these in smaller quantities. Now, the chemical composition of bacteria, if you take, it's more or less that of the higher forms of life, the eukaryotes. Like protein is about 55%, and RNA, DNA, phospholipid, lipopolysaccharide, murin, and inorganic ions. Murin is one item which is unique to the bacteria. So you do not find it in many of the life forms. And what are the nutrients? There are macronutrients and micronutrients. Macronutrients are those which are required in large quantities, say the carbohydrates, the carbon, hydrogen, oxygen, nitrogen. These are required in larger quantities because most of the organic substances have these ingredients. Whereas the micronutrients, they are the ones needed in smaller quantities. Like the manganese, zinc, all this is required in smaller amounts, especially for the enzymes, for pigment production, etc. And there is one category of bacteria called as the fastidious bacteria. The word fastidious means something like fussy. These bacteria do not grow under ordinary circumstances. They need to have certain special ingredients that have to be added if you want to grow them in the laboratory. So they require some complex nutrients or some items called as the growth factors, just as we humans require vitamins and certain amino acids, specific amino acids. So also there are some bacteria which are fastidious and they require some special features. Let's see what they are. Now, this is a chart showing the different life forms divided based on the energy source. So if the source of energy for an organism is light, then they are called as the phototrophs on the right side. Phototrophs, and if the carbon source is from inorganic compounds, you call them photoautotrophs. That means just by using carbon dioxide, some of them make carbon dioxide and light, they make their own food, as we've learned in our biology classes. So they are the photoautotrophs. And then if they require organic compounds and light, they are called the photoheterotrophs. We will not go more in detail in this category because the bacteria that we are interested in, the medically important bacteria, are all in the other side. This is the chemical or the chemotrophs, which require a carbon source in the form of an organic compound. And they are the chemoheterotrophs. Heterotrophs because they require chemical source and an organic source of carbon. Therefore, they come under chemoheterotrophs, where the final electron acceptor is oxygen. So most animals, fungi, all the eukaryotes, the bacteria, they all come, the medically important bacteria, all of them come under the chemoheterotrophs. And this is what we will be talking about today. So energy and carbon both come from organic compounds in the case of chemoheterotrophs, and the same compound can provide both. Specifically, the energy source is electrons from hydrogen atoms in organic compounds. And there are two categories of living beings here, those that live on dead and decaying matter called as the saprophytes, and the parasites which require nutrients from a living host. So this again, you must be knowing from your biology classes in school. So we'll be talking about the parasites, which are the bacteria. This group, they include most of the bacteria as well as the protozoa, fungi, and other higher animals. So the chemoheterotrophs are the microbes of medical importance. Now, what are the growth factors required? Some bacteria require certain organic compounds called as growth factors. I told you they are also called as the bacterial vitamins, just as we require vitamins and minerals in smaller quantities in addition to the carbohydrates, fats, and proteins that we require. So the growth factors could be of two categories, essential or accessory. If it is essential, the bacteria will not grow unless you provide that growth factor. An example here is the X and V factors in Haemophilus influenzae. Here, I just want to caution you all that this is X and V factor. Please don't say this is Roman number 10 and 5. It's not 10 and 5, it's X and V factors. I find many of the students in the exams writing it as, 10 and 5. So X and V factors are required by Haemophilus influenzae, which is one of the bacteria. And these are essential growth factors. And the accessory growth factors are those which enhance the growth of the bacteria, but they are not absolutely necessary for it. So the accessory growth factors are something like our vitamins. So just as the eukaryotes require vitamins, the bacteria also require some vitamins. And some of these vitamins can be from our own vitamin B complex like thiamine, riboflavin, nicotinic acid, pyridoxine, folic acid, and vitamin B12. An example here is that vitamin B12 is used by lactobacillus, which is one of the gram-positive bacilli. Now, in microbiology, examples are very important. They may ask you in the Viva also to give examples, and in theory also you may have to give examples, because that indicates that you have understood your topic and you know what categories, what bacteria comes under which categories. For example, they may ask you, give examples of aerobic bacteria, give examples of gram-positive bacteria, give examples of gram-negative bacteria. So everywhere, examples are very important in medical microbiology, especially for your exams and also for your Viva. So factors affecting bacterial growth, the availability of certain nutrients, availability of water, the proper temperature, the atmosphere, that is oxygen and carbon dioxide, how much of it is present, and pH, moisture, drying, osmotic effects, radiation, mechanical and sonic stress. All of these are important for bacterial growth. So we'll take them one by one as per their importance. The temperature, varying temperature requirements are there in different bacteria. Some of them grow at high temperatures, some of them at low temperatures, but optimum temperature is when the growth occurs maximally and the best growth occurs. Usually, for most of the bacteria which are of medical importance, they grow around 37 degrees centigrade. Why? Because our body temperature, that is the human body temperature, is around 37 degrees centigrade. So if they have to infect our bodies and grow and thrive in our bodies, they have to have that as the optimum temperature. So they should be capable of growing at that temperature. And what is thermal death point? It's the lowest temperature that kills a bacterium under standard conditions in a given time. Okay. So the different categories of bacteria based on the temperature requirements. The psychrophils, psychro means low temperature and phil refers to loving. So those which love low temperatures, they grow around 10 to 20 degrees centigrade. And the mesophils are the medium loving bacteria, that is temperatures between 10 and 48. And thermophils are the ones which love high temperatures. So they grow around in 40 to 72 degrees centigrade. And there are even some hyperthermophils. They grow at very, very high temperatures beyond 100 degrees centigrade. There are bacteria which can, I told you initially, there are bacteria which survive in all kinds of temperatures. So this is an example of the bacteria and archaea which live in the hot springs in the geothermal, heated by the geothermal reactions. Like in New Zealand, you have these hot springs springing up here and there. There also some of these thermophils can grow. Why it is important is, please see on the right side, I've given the Taq polymerase as the enzyme. Taq polymerase enzyme is found in one of these thermophilic bacteria which grows at very high temperature, and this is the enzyme which is used in your PCR reactions. I'm sure all of you know what PCR is, because thanks to COVID, everybody knows, even lay people know about the RT-PCR. So PCR requires the high temperature which is provided by this bacteria, that is the Taq polymerase. So how does optimal growth temperature relate to our daily lives? Why should we know about which bacteria grows at what temperature? Okay, so that's because the rapid growth of bacteria happens only around that mesophilic temperature. So that is the danger zone as far as humans are concerned. And the high temperature, you should know what bacteria, how bacteria gets destroyed at what temperature, so that when we use our autoclaves and other methods to sterilize surfaces or sterilize our, the culture plates and other items in the lab, you should know what temperature you have to use to destroy the bacteria. Okay, so for example, in the autoclave, we have fixed the temperature of the autoclave as 121 degrees at 15 pound pressure for about 15 to 20 minutes. Why did we arrive at that 121 degrees? That's because most of the bacteria, including the spore forms, they are all destroyed by about 120 degrees centigrade. So we increase that by about 1 degree so that we ensure that all life forms and all bacteria will be destroyed at that temperature. That's why all our autoclaves they function at 120 degrees centigrade. Okay, coming to the pH. pH, that is hydrogen ion concentration, again is important. A majority of bacteria grow best at neutral or slightly alkaline pH. That is 7 to 7.4. Why? Because again, this is because they are medically important bacteria which cause diseases. Therefore, if they have to grow ideally under our body conditions, our body pH is around 7 to 7.4. Therefore, the bacteria which are neutrophils, they grow best around these temperatures. And an example for this is Mycobacterium tuberculosis, which requires 6 to 8 pH. Then there are some bacteria which grow in acidic temperatures like the lactobacillus. And there are bacteria which grow at alkaline pH, that is about 8.4 to 9.2. The best example of this is Vibrio cholerae, which is the bacteria which causes cholera. It requires high pH. So why you should know this? Again, because when we grow it in the laboratory, we have to know what pH we have to have in the culture media. Coming to moisture and drying. Most water is very essential. I told you 75 to 85% of the bacterial cell is water. So if you dry the bacterial cell, it could be drying, could be lethal to the cells. So again, this is important because we should know at what, how to destroy the bacteria. So we have to know all their properties. Effect of drying varies. Like if you take Treponema pallidum, which is a very, very delicate bacteria, it's highly sensitive. So you cannot keep it under dry conditions. Whereas Staphylococcus is a more hardy bacteria, which can stand for a few months even if it is dried. And the bacterial spores, they are highly resistant forms. So they can withstand desiccation. Desiccation means drying. They can withstand desiccation and they may survive for several decades. So again, here there is a large variety. That is, some bacteria can tolerate moisture drying, whereas the others, they cannot and they are highly sensitive. And freeze drying or lyophilization is what we do in the laboratory to preserve the bacteria. So when you freeze dry the bacteria, they go into what is called as a suspended animation kind of a state, where the bacteria can be preserved for over 60, 70 years. And after you freeze dry them, when you thaw them and you add them to the culture media, they will grow. So the other important requirement for bacteria is osmotic pressure or osmolarity. Most bacteria require an isotonic environment or a slightly hypotonic environment for optimal growth. And there are bacteria which require high salt concentration. They're called as the halophils. Halo is because halogens. So salt comes under the halogens, that is sodium, chlorine. Chlorine is one of the halogens, isn't it? So halophils are the salt-loving bacteria. So again, here the example is some of the halophilic vibrios. They can grow in high salt concentration. So what is the effect of salt on the cells? So this is the first. On the left side, you can see a normal cell that is in isotonic solution. That means the concentration of solute or salt is equal on both sides, within the cell as well as outside the cell. In the second, on the right side, you can see that if the concentration of the solute, such as sodium chloride, if it's higher in the surrounding medium than within the cell, then water tends to leave the cell. So the cell will shrink and it causes plasmolysis. So again, here the same thing is shown. The first picture is isotonic. That means everything is equal outside as well as inside, the concentration of sodium chloride. Whereas in hypotonic solution, outside it is less and inside it's more. So the water will go out of the cell and the concentration of the solute is within the cell is higher. So again, water will come into the cell and the cell will swell. In the third one, the concentration of the solute is more outside the cell, and therefore from within the cell, the water goes out and the cell will shrink. And what about uptake of nutrients by bacteria? Here again, you have to know about the two types, the passive transport and active transport. So passive transport is, as you know from your physics, you must have learned diffusion. Diffusion is again two categories, simple diffusion or facilitated diffusion. Active transport is where as the name suggests, actively the nutrients are transported. Let's see how it is. For passive transport, there are certain, the most important thing is that it does not require energy. And the substances, they will just travel from higher concentration to lower concentration. So there should be a gradient for passive transport to occur. Okay, the examples are simple and facilitated. So on the left side, you see the simple diffusion where the concentration of the nutrient is higher on the outside than the inside of the cell. So it moves from outside to inside by diffusion. Whereas in facilitated diffusion, there are certain carriers to facilitate. Please remember, this is different from active transport because there is no energy involved here. Again, here from higher concentration to lower concentration, it goes, and there's no involvement of energy. Therefore, this is a type of passive transport only. Only thing is, it is facilitated because it's something like what is shown in this picture. It's made easy to travel from the higher concentration to lower concentration. Whereas in active transport, it requires energy and some carrier proteins. And one important feature is it is gradient independent. That means irrespective of which side has the higher concentration of the nutrients, they can move either way by active transport. So active transport, here on the left side, you can see it requires energy. Energy is in the form of ATP, that is adenosine triphosphate. So whatever the concentration on the outside, when you want these nutrients, if you use energy, it can be taken into the cell. So it's on the right side, you can see that there is an enzyme which facilitates the intake of glucose. This happens in bacteria. So it facilitates the intake of glucose across the cell, across the cell membrane into the interior of the cell, that is the cytoplasm of the cell. And this happens with use of energy. Okay, and here you can also see that the glucose is changing its characteristic because it becomes glucose 6-phosphate. So here the group translocation, that is the transported molecule, also gets chemically altered. So it may or may not be chemically altered. On the left side, active transport, you're just getting the active ingredients into the cell, whereas on the right side, they are not only the glucose molecule is coming into the cell, but it's also getting altered. So this again is one form of active transport. Now coming to radiation and stress, radiation like X-rays, gamma rays, they can be lethal to bacteria. So when you grow the bacteria, you have to be mindful of these factors. And mechanical and sonic stress, this again can rupture the mechanical stress, can rupture the bacterial cell and destroy them. Now, a very important aspect, the air requirement, the oxygen requirement of bacteria. So you have various categories of bacteria depending on the requirement of oxygen. So these are the various categories. This can be a question in your theory exams. You could be asked about this in your viva. So please remember the aerobic, anaerobic organisms are very, very important. This is an important part of the topic. Aerobic means the growth requires the presence of air or free oxygen. So the bacteria, an aerobic bacteria will only grow in the presence of oxygen. And if the bacteria is very strict, it's called an obligate aerobe. An obligate aerobe is a strict aerobe which will only grow in the presence of oxygen. For example, Vibrio. And an anaerobic bacteria is one which grows only or best in the absence of free oxygen. That means there should be no oxygen for it to grow. Again, here you have obligate anaerobes where even if a little bit of oxygen is present, the bacteria cannot grow. So it requires a complete absence of oxygen. Example, Bacteroides fragilis. Again, here, please remember when you write your answers in the exams, you have to give examples for every category. Then you have the microaerophilic. They grow best in low concentration of oxygen, for example, Campylobacter. Facultative anaerobes or aerobes. It means that it will adjust its metabolism depending on the oxygen concentration. So most of the medically important bacteria come under this category of facultative anaerobes or aerobes. So they will adjust themselves according to the presence of oxygen and grow. Example, Staphylococcus aureus. Then there are aerotolerant anaerobes. That means these bacteria are basically anaerobes, but they can tolerate or they can ignore the presence of some amount of oxygen. For example, Clostridium Botulinum. And there are capnophilic microbes. Capno refers to carbon dioxide. So these bacteria, they require 3 to 10% of carbon dioxide for growth. Example, Neisseria. Why do you have to know this? Because when you grow the bacteria in the laboratory and try to diagnose illnesses, infections, you will have to know what media to use, aerobic or anaerobic or microaerophilic or whatever. Now, oxygen related to growth size. Here you can see in this tube, in the same tube of liquid medium, you have aerobes growing at the top because it's closer to the air. And at the right at the bottom, the anaerobes are growing because there's no oxygen available there. And in between, you have the microaerophilic organisms. Similarly, here you can see the obligate aerobes grow towards the top of the test tube. If you're growing it in liquid media, you'll find that the obligate aerobes, they grow at the top, whereas the obligate anaerobes will grow right at the bottom. And in between, you have the facultative anaerobes and the aerotolerant bacteria. So oxygen, why is it toxic for the anaerobic bacteria? Because oxygen is readily converted into some radicals like singlet oxygen, superoxide, hydrogen peroxide, and hydroxyl radical. So these are very toxic to the cell. And most of the important detoxifying enzymes, most bacteria have the enzymes to detoxify these, the toxic radicals. So these enzymes are mostly superoxide dismutase and catalase. So depending on the presence or absence of these enzymes, the bacteria will be able to survive. So oxygen is reduced to hydrogen peroxide by certain enzymes, and it's also reduced to the radical oxygen by ferrous ion. And in aerobes, they have superoxide dismutase and catalase. Therefore, they will be able to, they will be able to remove these toxic chemicals. But the strict anaerobes, they lack these enzymes, catalase and superoxide dismutase. So when these oxygen radicals are produced, they are unable to remove them, and therefore they are killed or destroyed. Now coming to enzymes. Enzymes play a very important role in bacterial metabolism. They are biological catalysts which increase the rate of chemical reactions by lowering the energy of activation. And remember, the enzyme is not permanently altered in the reaction because it can be reused, the same enzyme, at the end of the reaction. And you have different kinds of enzymes. Simple enzymes, they are entirely protein. All enzymes are protein structures, but simple enzymes have only protein, whereas the conjugated enzymes, or holoenzymes as they are called, they have a protein component and a non-protein component. So the protein component is called the apoenzyme, and the non-protein component is called the cofactor. So the cofactor could be iron, copper, magnesium, any of these, or it could even be some organic molecules. So here is an example of the apoenzyme, or the protein portion, that is the blue part, and the coenzyme is the light blue portion. So you need the coenzyme in order for the enzyme to act on the substrate. The coenzyme is also required. Only then you get the whole enzyme, that is the holoenzyme. So again, types of enzymes are there in bacteria. There are two types, the constitutive enzymes and the regulated enzymes. Constitutive enzymes means these enzymes are always present in the bacteria. Whether the substrate is there or not there, regardless of that, these enzymes are being produced and they are there. Whereas the regulated enzymes, they are not always present. They will only be induced and produced only when the substrate is present. So only when the substrate is there to act upon, then these enzymes get produced. So this is in a way conservation of energy, isn't it? So you don't really have to have all the enzymes all the time. When you need them, you produce them. That is what the regulated enzymes are. So this is just a diagrammatic representation of the same thing that I explained, the constitutive and the regulated enzymes. So there is one enzyme which is very important in diagnostic microbiology, that is catalase. So it helps you to differentiate certain bacteria. So we use it as one of the diagnostic tests for identifying certain bacteria. For example, Staphylococci and Streptococci, both are gram-positive cocci. So how do you differentiate between them? You use the catalase enzyme. If it turns out to be catalase positive, then it is Staphylococcus. If it's catalase negative, it is Streptococcus. So we identify bacteria based on certain tests like this. Now, coming to the microbial metabolism. So we've done with nutrition and growth. So the last part is metabolism. Bacterial metabolism, it is the sum of all the biochemical reactions. Why should we learn about bacterial metabolism? Because we want to know how to inhibit or stop the growth of the bacteria. So we want to know how to control bacteria. The other reason is there is a unity of biochemistry. If we understand bacterial metabolism, it helps us to understand human metabolism also, because basically when you go to the molecular level, it's the same, whether it's human or bacterial. So metabolism can be of two types, anabolism and catabolism. As you must have learned in your physics and in your physiology classes, uh, anabolism is building up. So this requires energy. And catabolism is breaking down. This again requires energy. So how is the energy coming? That is by three biochemical mechanisms: aerobic respiration, by anaerobic respiration, or by fermentation. These are the three methods. So you cannot have anabolism happening without catabolism or vice versa. Both of them happen simultaneously in all of the cells. So you can see anabolism is using nutrients. The bacteria biosynthesizes everything, all the macromolecules, proteins, carbohydrates, lipids, etc. And once these are built, the bodybuilding happens, the macromolecules, and then they are again broken down for release of energy. So again, that is broken down into certain the waste products, that is acids, alcohols, whatever it is. So simultaneously, the catabolism and anabolism is happening. And the metabolic strategies, that is, I told you, three biochemical mechanisms basically: the aerobic, anaerobic, and fermentation. In aerobic, I won't go into all the details here because this is more of your biochemistry, where you must have learned all this in your first year. So I'm just trying to, it's a kind of a recap for you. Aerobic respiration, remember, the pathways are the same as anaerobic, like you have glycolysis, tricarboxylic acid cycle, and the electron transfer. All this happens. But here the final electron acceptor is oxygen. So it is aerobic. And here the maximum energy yield is there. That is 38 molecules of ATP you're getting, adenosine triphosphate. Whereas in anaerobic, you're getting a variable amount, but less than this. And anaerobic also, the pathway is the same, but the final electron acceptors are nitrate, sulfates, or carbonates. So it is other than oxygen. This is not as efficient as the aerobic respiration. So variable amount of energy is released, and fermentation is the least effective, where glycolysis, the pathway here is glycolysis, that is glycogen or glucose is broken down, and you get the final electron acceptors are various organic molecules, and the energy yielded here is just about 2 molecules in comparison. So ATP is adenine with the ribose sugar and 3 phosphate groups. That's why you call it triphosphate. And when you remove the terminal phosphate, it becomes ADP, that is diphosphate, and energy is released. So ATP, it contains the energy that can be easily released, and it is required for all the anabolic reactions. And aerobic respiration, as I told you here, oxygen is utilized and maximum energy is provided by this method. Anaerobic respiration, the final electron acceptor is other than oxygen. Like you have the nitrates in some bacteria like Pseudomonas and Bacillus. And in other bacteria, you may have sulfates, you may have, the carbonates and all this. In most facultative organisms and obligate anaerobes, this is the kind of, the respiration that occurs, anaerobic. And there is lower production of ATP because the complete TCA cycle is not performed here. It is cut short by these, the final electron acceptor, which is not oxygen in this case. And glycolysis is splitting of sugar. The carbohydrate catabolism occurs, and oxidation of glucose into 2 molecules of pyruvic acid occurs in this. And there is degradation of the carbohydrate through a series of oxidative reaction. And the end products here are pyruvic acid, NADH, and ATP. So ATP, you're only getting 2 molecules. So fermentation, which happens in most bacteria, is incomplete oxidation of glucose or other carbohydrates in the absence of oxygen. And it uses organic compounds as the terminal electron acceptors, and only a small amount of ATP is produced. And there's formation of acid, gas, and other products by action of various bacteria on pyruvic acid. So fermentation, the end products, the beginning is pyruvic acid, but the end products could be various other chemicals. For example, the fermentation of Streptococcus and Lactobacillus will yield lactic acid. Some of them will yield ethanol, that is alcohol. carbohydrate, carbon dioxide, propionic acid, butyric acid, and some of them ethanol with lactic acid and different acids. So why is this important? That's why. So, apart from bacteria being, they are not all bad. So apart from bacteria being causative organisms of various diseases in humans, please remember, they are also useful in many ways, because depending on the type of organism and what the substrate that is used and the enzymes that are present in them, you can get a whole lot of fermentation products, end products like alcohol, then your yogurt or curd and cheese. So all these that we enjoy are as a result of bacterial fermentation. So with that, I finish my talk. I thank you very much for your attention. If there are any questions, I can take them. Thank you.
Thank you so much, Dr. Sumudi. Thank you so much for an elaborate session today. Yes, ma'am, there are certain questions definitely have been asked, and so I'll just go one by one, ma'am. Ma'am, first question is, in case of bacterial growth in the liquid media, why do the culture look turbid from the viewpoint of optics? Should I repeat
I'm
it
not
now?
able to increase this. Can
Ma'am, I
I?
repeat it. In
How do
case,
I?
ma'am, in case of bacterial
Just a
growth.
moment. I'm unable to.
Okay.
Just one sec. Okay. Yeah.
Ma'am, I repeat the question. In case of bacterial growth in the liquid media, why do the culture looks turbid from the viewpoint of optics?
From the viewpoint of optics. See, turbidity is as a result of growth. If you have no bacteria at all, the liquid is absolutely clear. That means there is nothing which stops the light from passing through, isn't it? Whereas when there is a lot of bacterial growth, and I told you, bacteria, they multiply in exponentially. That means, the doubling populations occur. So when there is a lot of bacterial growth, it turns turbid because the light is no longer able to pass through the liquid, properly in a straight line. So the bacteria are hitting, the light hits the bacteria and gets deflected and all. Because of that, the turbidity is visible. This is what we are measuring, in fact, in this spectrophotometer and all that. I told you the turbidity nephelometry is the study of opacities. So that's how bacteria are they can even be counted using certain machines and all that. They can even be counted this way by seeing how much of light scattering occurs and all that. So that's why the turbidity occurs.
Okay, ma'am, the next question is, I'll just put it up. Ma'am, in case of certain microbes which have longer generation time, we cannot provide the reports very soon, and we have to wait for a month. So what is the alternative for this? Can we go for biomarker testing?
Yes, that's a good question. Let's take, for example, the Mycobacterium tuberculosis. Mycobacterium tuberculosis, which causes tuberculosis, it grows very, very slowly. In fact, I have one of my research topics was that when I was a pool officer, and I used to grow it on the LJ media. The typical media that's used for Mycobacterium tuberculosis is LJ media, Lowenstein-Jensen media. So when you grow on LJ media, it takes anywhere between 4 to 8 weeks to grow. So we used to tell the patient, you come back after a month or so, and then only the growth can be, we can diagnose it and all that. But now what we have with all these newer molecular testing coming up, what we do is the DNA or the nucleic acid of the bacteria is what is detected. So if the bacteria, for growth to occur, it takes a long time because the colonies have to form. And one colony means thousands and millions of bacteria which have to gather together. That's what forms one colony, isn't it? So a visible colony requires multiplication 10 to the power 8, 9 times. Whereas if the bacterial DNA or the nucleic acid is present, that can be picked up by these PCR and other tests. Now you have the gene expert, which works on this principle, that is molecular testing, where the nucleic acid of the organism is picked up, and that's how it is diagnosed. So that makes it very quick. So the molecular based tests, and nowadays you have what is called as the point of care tests, POCTs they are called. So in these point of care tests, at the bedside of the patient, you can do the testing, and the result is available within about 20 minutes to about 30 minutes, half an hour. So if you can get your results that quickly while the patient is still in your clinic, they can just wait outside sitting for about half an hour or so, and if the results come, you can straight away start the treatment. And so these are the this is how advanced the diagnostic microbiology is today. Although, see, you cannot do away with these, because for research purposes, the culture and this, prolonged testing and all of that, they also may be important, because there are some antibiotic resistance and all of that. You still require culture for that. So you cannot do away with them, but your diagnostic procedures are hastened much because of all these molecular tests. So you don't really have to depend on even the slow growing bacteria can be detected by the newer molecular tests and the point of care tests. Yeah.
Ma'am, another question is, as temperature has an impact on bacterial growth, is hot food or cold food items also interfere with the growth of certain bacteria?
Yes, of course. Hot food, that's why they say, all these Staphylococcus aureus, let's take the example of this bacteria, Staphylococcus aureus and botulinum, this Clostridium Botulinum, these are certain bacteria which can cause food poisoning. So if these, they produce certain toxins in the food. So when we eat food which has these bacteria which have grown and produced toxins, we get food poisoning. Okay. So by heating up the food, these bacteria are destroyed. The vegetative forms of the bacteria, they are destroyed. So you can safeguard your food that way. And also by even they say, like, ice creams and all that. That's why they say ice creams, you are eating it cold, but it's better to eat it cold when it's fresh. If you store it in the fridge and then, repeated freezing and thawing, that's not good, because the bacteria in them, they can turn, they can produce the toxins, and they can multiply and cause some infections like food poisoning and all that. So all this Salmonella typhi, the one which causes typhoid, the bacteria which causes typhoid, so they can all grow and multiply in foodstuffs, like in milk products, like ice creams and all of that. So definitely heating the food has a role, because bacteria can be destroyed by that process. By the
Ma'am, a
cooking
similar question
process, you can destroy bacteria.
thanks, ma'am. Ma'am, so similar question is like, is there any role of a hot water or lukewarm water intake with the bacterial growth in human body?
I'm sorry,
I think it's a similar question that I asked. That is there any role of a hot water or lukewarm water intake with bacterial growth in the human body?
Yeah, some of the virulent bacteria, when you take hot water, they are destroyed. See, most bacteria are destroyed around 55, 56 degrees, which is like the warm water. So boiling hot water, definitely it would be more detrimental. But most bacteria are destroyed around 56 or so. But we still use 121 degrees in an autoclave, because the bacterial spores, they are not destroyed easily. The bacterial spores can survive at very high temperatures. That's why we fix the autoclave temperature as 121. But most bacteria many of the pathogenic bacteria are destroyed around 56, and which is your warm water or hot water. So at that temperature, many bacteria are killed, but some pathogenic bacteria can survive if they are sporulating types.
Okay, ma'am. Ma'am, another one is, ma'am, what is the significance? What was the significance of counting bacteria in an medium?
See, bacterial counting, this actually, when you are doing research and your studies or something, research work, then it becomes relevant. But otherwise, bacterial counting, as far as the applied aspect is concerned medically, is as far as I can see, it is in the this water count, like drinking water. The bacterial count becomes very important there. So if you go to a water purification plant, there the count should not be beyond the E. coli count. E. coli is Escherichia coli, which is a bacteria which is present in the colon, and it is there in the human, it is there in plenty in human feces and all that. So if you get this bacteria in a water source, that means the water source is contaminated with fecal contamination. So in such places, the bacterial count becomes important. It's called as the plate count. So the water source from such source, from a river or from a well or whatever, the water is taken and then you plate it, and then you count the number of colonies, and then you have certain calculations you make, and then you can see that if it's beyond a certain number, then you say this water is not fit for drinking. So it's called potable water. Potable water is water which is fit for drinking. So if it is not potable water, that is when the count is very high, then you say that it's not. So this is one field where the counting of bacteria becomes relevant.
Another question is, I think it's quite interesting one, that is that if we have an unknown bacteria and have tested it against various biochemical tests, can you please suggest a standard source or a book, a reference book, from which we can compare the results to identify an organism?
See, bacteria are, if you read your taxonomy chapter, which is your in your one of the first chapters in your microbiology textbook, in taxonomy they say that the bacteria, newer bacteria are going on being discovered, and they are all listed in that Burgess manual. There's a Burgess manual of systematic bacteriology, where all the bacteria, the newer bacteria, and all the bacteria in the universe which are known are all listed over there. So if there are certain centers in the world, like in USA, you have the in Bethesda, that is in Maryland, there is the NIH where they have all the known bacteria cultures maintained. They're called the ATCC strains. That means American type culture strains. So these ATCC type culture collection, sorry, ATCC is American type culture collection. So, and similarly, you have in Europe, you have the CLSI, they maintain the NCCLS strains. They have the cultures of most of the known bacteria are maintained over there. So when you want to identify a new bacteria, you have to go through with all these, you can use these control strains or the standard strains, and then try and compare if any of the biochemical reactions or something are matching with these bacteria. If not, then it is a new bacteria, and then it will have to be, whatever the properties are, that will have to be listed, and then it will be listed in the Burgess manual. So as of now, the number of bacteria that are there, which are medically important bacteria, most of them are the known ones. And what we isolate are mostly the known bacteria. So when we do the biochemical reactions, we know what to expect. Like if you say Staphylococcus aureus, it should be catalase positive. Okay. If it is catalase negative, then you can rule out safely, you can rule out Staphylococcus, then it should be Streptococcus. And under Streptococcus, which type it is, again, you do different biochemical tests. So there are a whole lot of biochemical tests and a whole battery of tests for identifying the bacteria. So most known bacteria, which are of medical importance, can be identified. Now you have even the automated machines for identifying this. You don't really, all the biochemical tests are done by the machines, like the Biomero's Vitek and all that you have. So you just have to put in the sample and it'll just give out the result, the species of the bacteria, because all the battery of tests are done in the machine. But in the laboratory, manually also, you can do the various biochemical tests, and that's how they can, you can identify the bacteria.
Ma'am, second last question, is it maybe third last? I can say I'll just another 5 minutes of yours. How can we prevent food? That's the general question. How can we prevent food from cold spoiling bacteria in refrigerator?
Prevent.
Yeah.
So
how can we prevent food?
that is the normal precautions that you take. See, when the food is hot, you keep it. You don't keep it in the fridge when it's hot. So when it's hot, you assume that there are no bacteria in it. So that time you have to protect it by covering and all of that. And then you store it in the refrigerator. So, and your refrigerator should not have any of those bacteria. So the refrigerator obviously has to be kept clean and, sort of sanitized and cleaned every now and then. So pathogenic bacteria are not expected in the refrigerator of normal, if it's normally kept clean and all of that.
Ma'am, second last, ma'am, can you please again explain the O2 toxicity?
Oxygen toxicity. Yeah, see, when oxygen is getting into the cell, into the bacterial cell, during the metabolic process, the oxygen is converted into certain radicals. As a result of many the metabolic reactions, oxygen is converted into certain radicals. That is the some of the radicals I did mention, hydrogen peroxide. It will get converted to hydrogen peroxide, and then the catalase enzyme has to be there to remove this hydrogen peroxide and to convert it into water and oxygen. See, H2O2, it will be converted to H2O plus oxygen. So oxygen is not lethal and H2O is water that again is required in the body. So when this important enzyme, catalase and the catalase enzyme and superoxide dismutase, these important enzymes, if they are present in the bacteria, then it will be able to remove or detoxify the radicals of oxygen which are formed. But if these enzymes are absent, as it is in the anaerobic bacteria, so such bacteria, if you put them in oxygen or if you provide oxygen to such bacteria, they will not survive because in the metabolic process, when these toxic radicals of oxygen are produced, they will not have these enzymes, catalase and superoxide dismutase. They don't have these enzymes to break down that toxic radical of oxygen and make it into non-toxic water and oxygen or whatever it is. So they will not be able to do that. So it becomes toxic to such bacteria. For example, the Clostridium tetani is one of the anaerobic organisms. Clostridium tetani is the bacteria which causes tetanus disease. So this bacteria is anaerobic. So you cannot grow it in the presence of oxygen because it lacks these enzymes, catalase and superoxide dismutase, to convert the radicals which are formed from oxygen, to convert them into harmless water or oxygen molecules. It lacks the enzyme. So it will naturally get destroyed.
Ma'am, another question is, ma'am, are the sanitizers actually useful against microbial biofilms?
For biofilms, some of them are, but films by and large, they are nothing but a group of bacteria with a covering of, a capsule. So if that can be penetrated by the sanitizers, then it becomes useful. So some sanitizers contain, it depends on the chemical constituents in the sanitizers. And there are ways of destroying the biofilms also. So they are susceptible to some of these chemicals.
Ma'am, it's saying a little bit introduction about botulism.
Botulism is a whole chapter actually, the Clostridium botulism. So Clostridium botulism is caused, it's a kind of food poisoning, and it is caused by the bacteria which is called Clostridium botulinum. You'll read more about it in the chapter on Clostridium. But just briefly, let me tell you, it's one of the anaerobic organisms which grows under anaerobic conditions. That means oxygen should not be there. And in children, there is, they say even honey or something, if it is contaminated in children, it can cause botulism. And it causes food poisoning. In fact, I think if you see some of the textbooks, Indian textbooks, they give that the fried rice, the Chinese fried rice, which is not totally cooked. Of course, it's very tasty, but then it's not totally cooked many a times. So if it is contaminated, you can get this botulism or type of food poisoning. So these are this if Clostridium botulinum is present in the foodstuff, it's basically a spore forming bacteria. So if the spores are there in the foodstuff, so when it is at appropriate temperature, the spores will germinate. Then the bacteria is released and it produces the toxin, and the toxin is what causes the symptoms that you get, like vomiting or diarrhea or whatever. So that's just in brief, but please read the chapter to know more about the bacteria and the kind of food poisoning it causes.
Ma'am, just two more questions, the last two questions only. Can biofilms be seen in our body?
Yes, they say more if there are some people who have had these, any prosthetics, like people who are using the valves, artificial valves, or some prosthetics in the form of orthopedic, these prosthetic surgeries who've had. So, and even your the dental thing, any artificial inanimate thing that you introduced into the body when we use it in the form of prosthetics, you know, some of the artificial valves, then orthopedic prosthesis. So these, like all those steel plates and all, which they put sometimes in fractures and all that. So such places, if they get infected, the bacteria can form a film on these inanimate surfaces and cause biofilms can be produced that way.
And the last question, I think it's a very interesting one. Is Ms. Shinde is saying that is a very insightful talk today, but what she's asking is, what do you think is the future of microbiology research? Like, is AMR or antimicrobial resistant the only hot topic, or there are any other topics too for research or for PhD students?
As far as microbiology is concerned, the sky is the limit. So you think about it and you can do it. So whatever field it is, whether it is antimicrobial resistance or something, that again is an important one. But apart from that, just any part of microbiology you take, any topic, one can always do research, and it will always be interesting because the microbes are very interesting. And although we know so much about it, there is a whole lot more, believe me, which we don't know about it. So if you want to do research in microbiology, please go ahead by all means and do it, because as I said, the sky is the limit.
Well, thank you so much, ma'am. Thank you for taking all the questions that we have today and always
Yeah,
stretching us
thank you
in by
very
the next
much.
20 minutes. So I thank you, ma'am, for being part of today's session today, and also to all our participant attendees in the session. And as far as from Walters Kluwer, definitely we are going to provide more and more such sessions for the in the future for medical students as well as faculties on the medical and the newer topic that we have. Thank you so much again, ma'am, for your time today.
Yeah, thank you very much. Thanks to all of you for your patient listening. And thanks to Walters Kluwer for organizing this. Thank you very much.
Thank you, ma'am. Thank you so much.